Hi everyone, and welcome to the eighth TIA Centre seminar of the year. So my name's Adam Shepherd, and I'm a researcher here at the TIA Centre. For those that are new to our seminars, we invite researchers and leaders in the areas of computational pathology and beyond to present their sort of leading research. I'm also have a co-host in Jachi now, who has helped set up and organise this particular seminar, and also Kezi helped as well. And today it's a real pleasure to introduce Dr. Yang Hu, who is here today in person. Yang is a lecturer at the School of Computing and Mathematical Sciences, University of Leicester, having ended his contract as a research scientist at the Nuffield Department of Medicine at Oxford. And his research focuses on computational pathology, medical image analysis, and the development of lightweight and interpretable AI methods. And the title of Yang's talk today is Computational Pathology Before and After the Foundation Model Era. So with that, I'll pass over to you, Yang.
Okay.
And just to double check, do you mind if there's questions on the chat on the channel where I pose them to you, or would you rather wait until the end for us to ask any questions? It's up to you.
After that? After in the end?
Yeah, cool.
Yeah,
That's
in
fine.
the end,
So just a reminder to everyone online, if you've got any questions, please feel free to post them on the chat, and we'll answer them at the end. So thank you, Yang.
Okay, thank you. Is it everybody can hear me?
Yes.
Okay, and well, it's quite be honored to be here and be invited to give this talk. Actually, although I never been working in this brilliant lab for even 1 day, but I have a lot very old friends with you. So basically, I think I have a very close connection with you and so today I just would like to have a very flexible conversation about some of my feeling and my experience especially in the last few years when I was doing something computation pathology research. It's a very game-changing era in recent years. The pathology foundation model just changing everything. So I would like to just share some feeling about that. So we have this topic, and so about myself, I actually Adam has already give a very nice introduction about me. So uh, I don't have to say too much more, but I have a lot good friends with your like Kausuk and like Daoud. I have mentioned. And the group leader in this in TIA lab also have a very good relationship between with my old with my previous lab in Oxford, and we have a lot of collaboration already. So I just assume I just assume everybody in this room is also online familiar and very well about digital pathology. So I will quickly skip these slides. So about the how we get the digital pathology, how we get from biopsy, sectioning, scanning yet, and digitalization that for sharing and computing on the computer, and I also assume you know very well about how is the main challenge of the digital pathology image analyzing. The whole slide digital pathology images. It's very high resolution, and usually we need to separate it into a lot normal size images and to suit for our computational resource now. And the multi-instance learning we know is a very typical practicing we are using to deal with the whole slice image, and also because we don't have too much resource to ask the pathology help us to label and to help us annotate the every pixel, every detailed pixel. Maybe you have that resource. I have already saw a lot of work from TIA. You have a very strong resource to ask pathology to help you annotate even each cells. So that's you get very good. You have get a lot very good works on some cell segmentation or something else. So, but usually we don't have that resource. So we use that weakly supervised paradigm called multi-instance learning to predict to share one predictive label in for the whole slide. Um, so the first work I would like to introduce is before the foundation model area that so during this work we have this very special setting up that is we would like to use the histomorphological features to do some molecular subtype prediction. So as we know, the tumor subtypes based on the molecular choice just can indicate us very precisely on the personalized treatment for different group of patients. Uh, but it's highly rely on the sequence analyzing equipment and it's very expensive. They're relatively more expensive than just looking the histopathology images. So 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 that's why we need to find a cheaper alternative way from the perspective of the digital pathology image analyzing. So basically, that's a very naive uh, DAO class classification task. It's just so. For example, we have a molecular subtype named KRAS. In colorectal cancer, we just need to train classifier to perform that classification into yes or no if that's KRAS or Y type. Um, so a very pioneer work about in published in about 5 or 6 years ago, by which conducted by our old friend Kosuk, uh, is just raise that question: is that possible to identify the morphological correlated that allow us just to predict the molecular subtype on colorectal cancer only based on the histology images, and they just raised that question very early, and they conducted such an experiment and find that, and just training such a very straightforward classification pipeline and get very good result even on the external validation cohort. The result looks quite good. So that just a proof a thing is only use digital pathology images can conduct that uh, can conduct that prediction about um, different molecular subtypes. Mm, so the whole pipeline just be um, constructed very successfully. So we would like to expand this idea, the similar idea, to the other type of the cancers, like one of our collaboration with John Radcliffe Hospital in Oxford. We have a cohort on ovarian cancer. They are very interesting on prediction of the epithelium muscular transition EMT. We call it ENT. So basically, this molecular subtype can be separated into high or low, and the EMT high tumors account for about 20% of the serious ovarian cancer, and which is highly associated with the poor survival rate. Um, so, but the problem, and we even have some proof of concept work uh, has been published by the other people in our collaborative group. They have published some proof of concept paper on cancer cell and clinical cancer research, and but the problem is compare with the existing at that moment, compare with the existing method, all based on at least more than 1,000 whole slice image, but we only have 70 whole slice image on ovarian cancer cohort for our experiment. And so basically, if we separate the training and test set, we only have less than 50 of them for training. So actually, that's a very common issue we usually will face, especially especially we don't have enough resource to collect such a big cohort, right? Um, so that's why we just have a just have proposed another method. So before I introduce my method, let's just have a quickly review about typical multi-instance learning histopathology analyzing pipelines. Basically, the typical multi-instance learning just extract the high-level features from the pre-trained encoder, and that pre-trained encoder is weight fixed and cannot be optimized anymore. So the only trainable only trainable module in that pipeline is the attention-based aggregator. And of course, we can assign the different attention value on different tissue areas that just highlight which area is more interesting with the respect to the final predicted result outcome. But that's quite rely on large amount of data. If we don't have large amount of data, so such a um, even we just use a very slight network to be that aggregator, we cannot achieve a very good generalization. And we cannot further optimize the high level features anymore. There's no chance to further optimize that high level features. So that's why we propose that a very tricky one, self-interactive multi-instance learning require interML. So basically, that's a very simple idea contained three steps. The first is just still select the high attention types from the typical attention pool network. And then we just fine tune the high level feature encoder with those high attention types with the same prediction label. And now we have the optimized encoder, right? So we can refresh the high level features for the next round of attention pool aggregated training. So NEC is also collaborated with our old friends COSUC. uh, so this work was just published in last year, the medical image analyzing, and so we have a very good result. We get a very nice result, very nice performance improvement on four molecular subtype tasks, which include ovarian cancer, colorectal cancer, lung cancer, and breast cancer as well. And but it's not the most important result. I have more interesting results shows here. Like with each round of interaction, we can see that attention map is just moving from those noisy area to some more biologically meaningful areas and those 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 ties with the significant uh, biologically meaning will generally get more attentions after each round's optimization. But for those noisy area, maybe initially they have some highlighted color, so they gather more attention. But generally, after several rounds of interaction, the attention of assigned on them has been reduced very significantly, right? And we also check the feature space, and the feature space that means the informative the feature space of those informative tiles from different subtype was separate more significantly so that we can provide a easier distinguishable feature space for the aggregators classify. So that's why the classification the performance just being improved such a lot. Uh, so basically, with less than 50 training samples, we can achieve a very good regions of interest in detection capability, while the other method usually leverage more than 1,000 slides. Uh, so so now so that's a work just before we have the foundation model. Uh, for now we have a very data efficient approach and very data efficient approach in case we only have a very seriously limited training cohort. And basically the data requirement just from 1,000 be reduced to less than 100. And we can also provide very good expandable presentation at different scales. But I think a very important takeaway is that when we fine-grain the features are when the fine-grained features are optimized further, it shows very strong impact on the overall interpretable performance for the whole slides, for the whole area of tissue. So now we get into the second stage of our today's talk. Is the first I would like to start out with the question that does the higher accuracy really mean better? Is there something about the concern? Is the origin from some concern raised from the heterogeneous in AI model interpretability? And as we know, so basically in last 2 or 3 years, since C transpass, since C transpass is rubbish now. Uh, uh, more than 20 pathology foundation model have been introduced uh, and has been introduced, and every one of them just state they have a bigger and bigger cohort for training a stronger and stronger foundation model, and but they still have some they still have some relatively dangerous aspect that is um, most of them rely on very biased data source because most of them are training based on not only TCGA but also their own private cohort. Right? So their private cohort must most of them cannot. So as far as I know, none of them have such an organization capability to collect all the data around the whole world. Right? So basically, they can they can control one or two local hospital, but they cannot control every hospital around the world. And so most of them just rely on very biased data source. And another thing is, although every one of them just state they have their foundation model pre-training resource include every type of cancer, but the distribution of cancer types are super imbalanced. If you carefully check the paper of them, you will find that uh, their cancer type distribution is very imbalanced, right? So okay, they get too much focus on the accuracy, and they indeed get very good performance on those benchmark on those public benchmark, but we find that the stability and the reliability of their expandable result is not so good. I will show you more result here. So in this very in this actually we just did some proof of concept work. It's very naive work, just been submitted to a journal course. It's from Springer Nature, but not Nature. We just tested two very popular pathology foundation model on 5 downstream tasks, and uh, we did some set of comparative experiment, and so we first of all we found that these foundation models perform all very similar. The performance on the perspective of currency, AUC, area of curve, area under the curve, all these um, all these two foundation model performance is very similar, almost the same, and all good, all much better than ResNet. Okay, but the thing is, when we tracking the attention moving behavior to simulate the thinking trajectory of these foundation model during they transfer their knowledge existing during they transfer their existing knowledge on those downstream tasks, and we also check the attention influx and efflux areas, so that we also identify the region with the significant local attention changes. So we find that when we just checking all these attention moving behaviors, we we saw significant heterogeneous between these two different foundation models. First, we just reviewed the patch with a significant attention moving areas. We just observed extremely tiny overlap in their distribution across different across these two foundation models. And we just check on three of them. We just check the attention moving result uh, on three downstream tasks based on our private cohort. The first, ovarian cancer treatment response prediction. This is not the private cohort. This is the public cohort, but just not so popular. Um, so we find that the cluster shows obvious. Some cluster shows obvious overlap, but while more attention moving areas display entirely different phenotypes. So, so such, so when we check the attention moving behavior, we will find that the consistency of the attention move in or move out areas morphological understanding across different foundation models is so difficult to reliably capture. So, um, and I would like to show you more example on another cohort, another task. This one is colorectal cancer based on based on the private cohort in Oxford, and it also the uni and geek class also shows remarkably different local attention shifted behaviors. While the union would like to more like to moving the attention to those more uniform protein-like materials, but the GigPath would like to have a very strong preference on those well organized gland-like structures. Uh, and another example is on the grading or inflammatory bowel disease colitis. Uh, this task is also based on one private cohort in Oxford, and so the union more likely to moving attention to those early or middle healing changes, like just starting to rebuild the tissue for those areas. But the gigapaths are more likely to moving the attention to those later stage or more prominent tissue regrowth. Uh, yeah. So actually, this paper will be released. Has been released on MedArchive. I can show you later. I can show you later. I will send the link to I will send the link to Adam. Maybe after this talk. So, and a more interesting thing is that even we combine all the different data set and then we cluster the attention move in and move out tiles, we will find that different foundation models shows even some offset trait when they assign the attention move in and move out clusters to each other. So basically we can conclude that although the different foundation model shows the almost exactly same accuracy performance, but the but they are going to moving the attention to the completely different directions. So that's what that's a phenomenon actually we don't want to see. If we would like to have a stable, expandable AI model, right? So we are start to think about how we can solve this question, how we can solve this problem. Very intuitively, we just explore the representation diversity and how the different feature how the feature of different foundation model can help each other maybe. Um, so as we know, the pathology foundation model have a very strong diverse on the model on the training resource and training algorithm and the model architectures. So this is very naive, very intuitive idea to maybe we can get more robust we can get more robust feature by combining multiple foundation models. It's right. So I think everybody can think about that. So basically, what we want, our target is just like uh, first we would like to efficiently get more robust feature. The second is we also would like to maybe we can have some interpretable feedback on the performance of this foundation model from different for on different different phenotypes. So actually, the structure of this ensembling approach is embarrassingly simple. Uh, so we just extract the feature from 6 different foundation models, pathology foundation models, and we just use uh, we use uh, we use the average pooling to compress them into a smaller dimension because we don't have enough computation resource. As each foundation model will usually have more than 1,000 dimension of the feature space, right? And after we compress them into a smaller dimension, we just concat them together. We just concat them together, and for each tile on each tile, we just use the attention gate to dynamically adjust the weight of weight of these features from different foundation models for interpreting the foundation models in performance on different tissue phenotypes. So first of all, the performance looks very good, and it's lighter and better. So we combine the combined feature actually just be located in the center of the 2D space among all different foundation model feature space. And the dynamic combined feature just outperform all foundation models original feature across the board, across different downstream tasks. Here I shows two different downstream tasks. And so basically we have the conclusion that maybe the foundation model high dimensional features is also highly redundant. Uh, and another table shows the excellent result on spatial transcriptomic gene expression prediction task. The combined feature also achieve the best result averagely. And the combined feature have stronger capability to distinguish various tissue morphological features, morphological phenotypes. So like we just use the k-means to cluster all the tiles from in each slice based on the features of different foundation model along with the combined feature. We will find that the combined feature can distinguish the different phenotype more sensitively than the other of original foundation models. And we can show some another example here. we shows another example here. We can observe another interesting thing is that the foundation model just from the same country, from the same or from EU or same country, American, they tend to produce relatively similar unsupervised tissue clustering results like Koch, Uni, and Roche. They all from North American, right? And Ash, Optimus Zero, and Finkel all come from EU. But the proof gigapath just incorporates some case from China. So that's uh, yeah, and a more interesting phenomenon we just observed is that when we check the foundation models, when we check the foundation model attention contribution across the different tiles that at different locations, we just use the color to highlight each of them. We just find that foundation model will consistently contribute most to a specific tissue phenomenon, tissue phenotypes. So basically, we just based on these observation, we can conclude what you can see that the foundation model just shows significant diversity in their performance, or say in their excellently represented tiles. So um, in a more frankly saying is that maybe the foundation model, each of these foundation model have their own tasty, right? This is more example on that ovarian cancer treatment response cohort, and this on panda cohort all shows the similar phenomenon. You see here, right? And I will show you more. So, um, in this case, we just visualize the the distribution of the foundation model attention contribution on different area of tissues. You will see that phenomenon show again. And we also did a very interesting test that we just apply. Okay, so we just applied the weight distribution of this foundation model as the feature. So basically, that feature space only have 6 dimension for classification, and we just feed it into aggregate classification on this ovarian cancer ovarian Bayford response dataset. We get not very good accuracy because it's just a 6 dimension, right? We get the occurrence, we get the AUC of 72% versus only 59% by the max pooling multi-instance learning baseline. So that's what. So that means only based on those pure morphological features, pure morphological features that indicate the pathology foundation model, their preference as the feature itself actually already carries some morphological meaning of morphological meanings, right? So and I think we almost there. uh, and similar phenomenon also be observed on the spatial transcriptomic expression prediction cohort. Uh, well, so I would like to end up my talk with some discussion because I noticed that a lot of people would like to raise some interesting questions like, uh, so we would like to uh, have some discussed like the lightweight device, the truly interpretable model versus performance-driven foundation model. Um, how far we still, behind? How far the first one still behind the second? Uh, so Actually, before we start our discussion, I would like to have share some of my thinking that is foundation model superpower actually actually bring a more urgent need for the reliable interpretability and debiased applications. And but many so now the foundation model is not only on high level, the slight level foundation model have also dropped uh have also even dropped the basic attention based. You know, the attention based explanation is not perfect, but they even dropped that to in favor of currency or they don't have the enough computation resource, but they would like to have a slice level foundation model. I think that's a very dangerous trade-off for clinical using. And so the interpretability debiasing and the model for precision medicine. Actually, I think this topic cannot be separately when we consider them. They are they are they are very highly associated, highly correlated research topics. And how far can the existing foundation model take us to our target about interpretability and debiasing, and also good for using. So I would like to end at here. I would like to discuss if you have some questions. Yeah. Thank you. Thank you.
Fantastic. Well, thank you for an excellent talk there, Yang.
Yeah.
And thanks again for joining us in person. So yeah, let's open up the Q&A and let's see if anyone in person or online has any questions or anything they want to say on the discussion points that
Yeah.
Yang has raised at the end of this
Yeah.
slide.
Where I can I just check you dialogue.
Say again, sorry.
I just check your dialogue here.
Yeah. So
Yeah.
so the chat here, there's yeah. So Afsan said on the chat that she had to leave, but thank you very much. So let's start with the questions then. So I suppose my first thought really was on the slide that you had when you obviously were combining lots of different foundation models. And I think the slide that you said afterwards actually did that. So you do some sort of pooling per foundation model, feature embedding, sorry. Then you stack them and you put them through your attention model. And then I think the next slide you said lightweight and I'm trying to, I don't know what the actual wording was.
The lightweight and performance is
So
good.
I suppose when you say lighter, what do you mean? Because it's of course there's 6 times the amount of compute in terms of foundation models. Is it
the
embeddings? Are they
yeah, the
smaller?
lighter. Actually, the lighter is just we can storage the much smaller feature compare with the original size of foundation model.
So this is after you've done some sort of pooling on
Yeah,
the
reporting it. For example, here we have the foundation model with the original size or with the original dimension more than 1,000, but when we express that into 64 dimension, actually, and after we combine them together, is totally 46. It's totally 384, right?
Got you. So
the 384 is
so the sort of the downstream model is much lighter.
Yeah.
The original sort of still having to run your slide through 6 foundation models.
Yeah. So it's also 4 or 6 times smaller than the original size, the feature space.
Yeah.
Right. Yeah.
Okay.
Yeah.
I'm just going to ask, did you do any sort of work to look at like if there's a minimal set of foundation models that you can include in that fusion to get most of the performance? Like, so that maybe if you keep just like virtual and uni or something, you get most of the benefit of the combination and the rest.
Yeah. You see, at least in this table, we compress the feature into 512, 256, and 64 even. But we find that you see when we drop the dimension of this feature space of this feature from this foundation model to 64, you can see the performance if we not combine them. the performance has been deduced significantly, right? So we decided not to do that further. Yeah, but is that do we need to have a more aggressive deducing of this feature size to make it as 8 or 4? Yeah.
I mean, did you? Yeah.
Yeah. I think we tried 32 and 16. Not good. So we decided to stop it. Yeah.
I guess the question was less about how aggressively do you go into reducing the feature size of your final kind of output, and more whether, because at the moment you combine the features from 6 foundation models, right?
Yep.
Did you look to see if maybe you can
Combine
use
more?
4 of them and
Oh.
get the same result, for example?
Yeah.
Or if, like, basically, if there's any foundation models of that you can throw away and still keep most of the performance.
Yeah. Actually, so this paper is just we know we only have a conference version, but we have a more comprehensive one. We would like to submit in some journal. We have more comprehensive investigation about how we keep, because some of the foundation model, they are more likely collaborate with each other, but some of the foundation model are not very good friends, right? Maybe. Right. So we also have done some more comprehensive investigation on this on this aspect, and to check how we have a more uniform, more widely applicable strategy to combine different foundation model in different specific scenarios. Yeah.
It'd be interesting, as you said,
Yeah.
in terms of the interpretability at the end, whether you could combine an American one with a European one with a Chinese one.
Yeah.
See whether.
Absolutely.
Yeah.
And I suppose my next question was going to be about interpretability. You mentioned the slide level foundation models at the end, and obviously they're becoming much more popular now because they're easy to use. You can just go straight end to end predictions. But of course, you potentially, I mean, you can still get the heat maps out right, but they don't necessarily mean anything.
Yeah.
So, what's your thoughts in terms of interpretability there? Is that, I mean, the way you sort of phrased it at the end was that this was just a bad method, and we shouldn't be doing that. But is there not? Should we not instead be looking at ways to add interpretability to these particular methods?
Yeah. Well, actually, a very recent that is not included in this talk, but a very interesting, very interested, a very recent work we just finished in last week is about, we even use a lot TIA modules to do the cell segmentation. And so what we are doing is that we using some graph neural network to distilling the foundation models, so that graph neural network have somewhat inherit some knowledge of that foundation model, but that graph neural network can expandable about how the different cells are associating, how communicating with each other. So we can provide some other format of interpretability result beyond the attention, right? So that work will be submitted very soon. Yeah. Yeah. So I think we had TIA. In TIA we have a very good, a very cool paper is about something is all you need. Do you remember that one? A very old one.
Color is all where is paper?
Sorry, that
Was it?
That was color is all you need.
Yeah, something is all you need. I think is the author is Graham.
Oh, I was just thinking that, yeah.
Yeah, that one. So
one model is all you need. One model is all you need.
Yeah.
Yeah. Yeah.
Yeah, actually I think TIA provide a very good, cell level interpretable result. I think it for me is a very good opportunity to collaborate further with you because I use the I use TIA a lot. Actually, I think the cell level or some more fine-grained microenvironment interpretation is much more important than just attention, right?
I mean, that's
Yeah.
sort of typically the approach we end up taking, isn't it? Sort of finding heat maps and then getting
Yeah.
morphological features in those
So
areas.
just a heat map is
Yeah.
obviously not enough.
Yeah.
Yeah.
Okay. And I also wondered what your thoughts were in terms of sort of we had a seminar about 12 months ago by Peter Bohr, and he did a talk on and he introduced a metric on sustainability and foundation models, and
Yeah.
sort of talked about how we shouldn't be trying to use as many foundation models to get 1%, 2%, 3% improvement in accuracy when it's the sort of consequences it might have for the environment in the long run. So I don't know what your thoughts are, of course, with these sort of models which are using more and more foundation models, whether we should try and, I don't know, slim them down slightly.
Yeah. Well, to be honest, I think because the foundation models is so powerful, right? So we can't help us to we cannot stop us to use it.
But I suppose my question is having is it worth, so Virtua 2, right? You've got 85% accuracy, and for then times in our compute by 6 and using 6 foundation models, we're going up to 87 or something like that. We get an extra 2 or 3%. I just wondering if it was just a
Can you repeat that?
yeah, I suppose just a question in terms of um, more of a comment about using lots of foundation models, I suppose, and whether 1% increases are worth it, essentially, or 2%.
Actually, I don't understand.
No, don't
I don't understand. I don't understand what
No, that's okay.
You say combine different foundation model versus just keep improve one.
Yeah, I suppose my question was around that if we're using more and more foundation models that are just getting bigger and bigger and we're using more and more of them together, then it's obviously we can't really deploy these down the road. So is it worth the sort of the 1% increase in accuracy that we're getting? It was more of a sort of just a theoretical.
Oh, it's not worthwhile. It's not worth. It's unfortunately we still seeing a lot of people in the community still working on developing some bigger foundation model and just achieve 1 or 2%. And that 1 or 2% improvement even not true. So basically, it's just overfitting some benchmark, right? So I have I have a collaborator. I have a collaborator from Hong Kong University of Technology. I don't know if you know them. There's a Chen Hao. Who is the M-Star's author. Is that
used to be here in Western Asia as a visiting scholar. I think that's they collaborate.
Yeah, okay.
They also collaborate with Simon as well.
Yeah. And they did some very brilliant work on just the compress the foundation model into very small, very light weight, and they even can embed it on chip. And the performance just be sacrificed a little bit, and they would like to collaborate with some hospital. When they talk with the clinicians, the clinician told them, "Oh, you give us a very efficient, a very quick model. That's good, but the most thing we care about is still the accuracy need to be improved. We can take the bigger consuming computational resource, but but we want the higher accuracy. Yeah, we want a more current model. Yeah. Maybe sometimes the understanding between the technicians like us with the clinicians is different. Yeah.
Do you have any more questions online or in person that anyone would like to ask?
Hi.
So you've looked into the performance of several downstream tasks.
Yeah.
I'm not sure I didn't quite get all of what the tasks are, but I noticed most of them are, some of them were like mutation prediction, like you said, KRAS, you said different kind of molecular
Yeah.
predictions. So from my experience, when I've looked at those, when I used accuracy AUC, because of the high imbalance between the positive and negative samples, you tend to get very high numbers in these two metrics. But when you look at, for example, AUCPR, you would have that metric would be really differentiating between what is good and what is not good predictor.
Yeah.
So if you're, let's say, if you're predicting them all as one class, which is a majority class, you would sometimes get 70% accuracy, or even AUC would get very high percent. But when you look at the other metrics, it would kind of differentiate. If you look at the AUCPR, you would kind of differentiate if the model is actually predicting it or not. So I'm not sure if you looked into that as an explanation of the scattered heat maps that you showed earlier. So maybe they are all just not looking at something very useful. They're just all predicting the majority class, and that's it.
Yeah, that's very common issues we will face. But in my experiment, in our experience, we already considered that, and in those imbalanced labeling situations, we always use balanced accuracy to account our results. So basically all the result reported is balanced. Yeah. So we already considered that problem. Yeah. Thank you.
About the example of the foundation model, I've seen a lot of it's an interesting work or direction of work, and I've seen, I think recently in Nature Communication paper, that does kind of the same approach of exempling the foundation models, and they're saying that it gives us a better prediction, I think it was on blind post. Not sure if you have seen that thing.
I don't know which one you are talking about, but yeah, if you can share that to me, I'm really appreciate it. Yeah, actually, a lot of people, as I said, this is very straightforward, is a very intuitive idea, right? So I think a lot of people will think about that. So it's not surprised.
Performance.
Yeah.
Obviously, the task is different.
Yeah.
But I haven't looked at the results section like very deeply because it's on a different field. I'm not working on glioblastomas, but I don't know what was published.
Yeah, a lot of people are combining foundation model. A lot of people are distilling, are transferring, are fine-tuning. People will do that. People will keep doing that. And still a lot of people are keep developing a bigger a bigger one.
My question would be like, do you think it would be task dependent, so they can work better on some tasks, but on other tasks they won't work better?
Yeah.
Is it easy?
Task dependent, yeah. So actually, so in the last day I leave Oxford. Before I leave Oxford, I talked with Professor Ian, and we discussed a lot about which one will be an interesting things we would like to explore further. And like I think it may be very interesting to explore some disease specific foundation model rather than use because the pan-cancer foundation model is just technically reasonable, but clinically pseudo setting up.
So the question is getting 10 or 15, 20,000 slides per specific cancer, is that enough to train a foundation model
But we can
for it?
construct that specific disease foundation model uh based on the existing foundation model. We don't have to discard all the knowledge we have already collected from those existing foundation models, right? The existing foundation models are already very powerful. So the thing is how we fine tune it, how we maybe transfer somewhat of their knowledge to the disease specific models and make the disease specific model can be training efficiently and better, right? So that's a problem.
I guess there's those things called auras, which are a great way of taking a generalist model and tweaking it in some particular direction. If you think it might be a cool thing to like have a big generalist foundation model and then train a bunch of auras for each specific disease, and you just kind of learn up the aura for the disease you're looking at, and then you suddenly have a model which is kind of tuned.
Yeah, true. So for example, we don't have to just make some focus on one specific disease. We can combine several high relevant disease. Like we can make a foundation model for gastric cancers, right? So which include the colorectal, which include the stomach, all this stuff. This is a consistent cohort. This is a consistent system in human body. That make more sense to clinicians when we talk with them. Yeah.
Sounds good. Does anyone else online or in person have any final questions before? I think this room's only booked till 3. So any final burning questions? No, in that case, we've got one from Azam online, or Azam, sorry, online, who's got their hand up.
Thank you. Thank you for a great presentation. I have one
Hello.
question, and can you hear me?
Hello.
Can you hear me?
Have you got a question for Yang? Sorry, Azam?
Can you hear me?
Are your speakers muted?
My speaker is muted.
Oh, yeah.
Oh, yeah.
Hang on.
Okay. Hello.
Hello, can you hear me?
I just talked.
Do you want to put, are you muted?
Maybe.
So do you
No,
want
I'm not muted.
Can you
I'm
hear
not
me?
muted.
your volume up then, sorry.
This one?
Hello?
Hello, can you hear me?
Yeah, I can hear you now.
Oh, thank you. Thank you for a great presentation. I have one question and one comment maybe. So my question is about this hybrid method on all these features extracted by different foundation model. I'm just wondering if you so any specific patterns on the weights of different foundation models in this combined feature set or not? I mean, did you see, for example, any specific pattern? Some foundation model have larger weights in that feature subset or not? It is one question. And comment regarding that compression model. I didn't get the compression method you used for decreasing the feature set, for example, from 512 to 64. But we have already some study that could compress the feature vector embedding extracted by the foundation model to very smaller actually sets using some other feature selection methods. So yeah, there are already some works that the smaller set of features can reach the same or sometimes even better accuracy or IUC on these data sets. I have some work is specifically on TCGA, in Panda, not the data set that you worked on, but for TCGA, we already have seen some more compact feature sets for classification. Yeah.
Yeah. About your two questions. The first one, actually true. The first one, my answer is yes, and we did some study on investigation of this performance of different foundation model has then if they have some consistent patterns. Yes, they have, and we already found it. We will release that preprint maybe in next 2 months. And so this is the first question. The second one, actually, your comments very insightful, very good. Why we don't use some more supervised or something more informative method to compress our features is that we don't like some people just suggest me to, or even you use PCA to compress these features, compress the whole feature space of this foundation model is even better than nothing, right? So, but the thing is, why I consistently use this non-parameter method to compress the feature of foundation model is that because any of this compression, if we use some PCA or something, that compression is based on that smaller cohort, much smaller cohort than the pre-trained cohort these foundation model have. So we don't want the feature compression process introduced any effect from this smaller cohort. We don't want to introduce any biased knowledge from the small cohort. We just would like to very straightforwardly to compress them. So maybe that's my motivation and understanding on that. So yeah. Do you think?
Brilliant. Well,
Yeah.
I think
Do you think? I think heard of.
Yeah.
Yeah.
Thank you so much for your explanation. Yeah, I got your point. I totally understand what you mean, but maybe more feature selection method instead of feature comparison like PCA might be more useful because still with all those PCA and as you said parametric methods we map those features to a new space, so with some I don't know assumption something like that. But with feature selection, we just select some features originally from actually those features that the foundation model itself generated. So we tried some feature selection methods, and yeah, we could get some good results on comparing these feature sets extracted by those.
Yeah, I agree with you. Actually, I think it's depend on different application systems, right? So I think if we would like to make some specific task have a better performance, that's a very good idea to conduct that way. Yeah. Thank you very much. Thank you.
Thank you.
Yeah.
fantastic. Well, I think we'll draw the seminar to a close then. So just a quick reminder to everyone that's here and online. Our next seminar will be Monday, the 23rd of February, so 3 weeks away. And we're going to have an online talk by Dr. Danielle Belgrave from GSK. So I hope to see everyone there. But thanks again, Yang, for a fantastic seminar.
Oh, thank you.
It's a really interesting discussion.
Yeah.
Yeah, thank you.
Yeah, I really enjoy it. Yeah.
Brilliant.